Our today's topic is plant mitochondrial electron transport chain. Photosynthesis and cellular respiration are biological processes in plants that are closely related. Photosynthesis that takes place in chloroplast uses light energy, carbon dioxide and water to produce oxygen and glucose. Cellular respiration uses this oxygen and glucose to produce water, carbon dioxide and ATP, which is a form of energy. And this process takes place in mitochondria. During cellular respiration, organic compounds are oxidized to generate usable chemical energy in the form of ATP. ATP, adenosine triphosphate. So adenosine triphosphate is a molecule that stores energy in chemical bonds and is the primary energy source for many biological functions in living cells. So the respiratory electron transport chain of mitochondria is at the center of this process. Now, listen carefully. Aerobic cellular respiration is made up of three parts: glycolysis, the citric acid cycle, and oxidative phosphorylation. Oxidative phosphorylation has two parts: the electron transport chain and chemiosmosis. First is electron transport chain, ETC, and second is chemiosmosis. So this mitochondrial electron transport chain is a series of protein complexes and organic molecules in the inner membrane of mitochondria that transfer electrons and protons across a membrane. The ETC passes electrons from NADH and FADH2 to protein complexes and mobile electron carriers. Coenzyme Q and cytochrome C are mobile electron carriers in the ETC, and oxygen is the final electron recipient. Next is components of electron transport chain system. The respiratory electron transport chain consists of 4 oxidoreductase complexes. Just look at this diagram. This is inner membrane of mitochondria, matrix part and intermembrane space, and this is electron transport chain. So this ETC consists of 4 oxidoreductase complexes. First is the NADH dehydrogenase or complex 1. Second is the succinate dehydrogenase or complex 2. Third is cytochrome C reductase or complex 3. And fourth is the cytochrome C oxidase or complex 4. This ATP synthase is also known as complex 5. In addition, electron transport chain of mitochondria also consists of 2 mobile electron transporters. As I already told you, one is cytochrome C and second is lipid ubiquinone. Now, we are discussing electron transport chain. So, from where all electrons enter the electron transport chain? So now, next is electron entry pathways into the ETC. All electrons enter the electron transport chain via NADPH, which are generated by a variety of dehydrogenases in the mitochondrial matrix or in the inner membrane space, or via flavin nucleotides, FADH2 or FMNH2, which generally are bound to proteins termed flavoproteins. So, 4 electron entry pathways into electron transport chain has been identified. 1, 2, 3, and 4. One is the matrix NADPH pathway. Second is matrix FADH2 pathway. Third is intermembrane space NADPH pathway. And fourth is intermembrane space FADH2 or FMDH2 pathway. First is the matrix NADPH pathway. This. Here electrons are transferred in the form of NADH to electron transport chain. NADH is reoxidized by complex 1 or the internal alternative NADPH dehydrogenases and transfer electron to ETC. Second is matrix FADH2 pathway. Here FAD containing enzymes oxidize carbon compounds in the mitochondrial matrix and directly or indirectly transfer electrons to the ubiquinone pool. Third is the intermembrane space NADPH pathway. Cytoplasmically formed NADPH is reoxidized by external alternative dehydrogenases and transfer electron to complex 1 of electron transport chain. Fourth is the intermembrane space FADH2 or FMDH2 pathway. Here, FAD or FMN containing enzymes oxidize carbon compounds in the mitochondrial intermembrane space. Then electrons are transferred either to ubiquinone or to cytochrome C. Now next is components of electron transport chain and the mechanism of action. So first is complex 1. So I'll repeat here. This is inner membrane of mitochondria, matrix, intermembrane space, and this is electron transport chain, different complexes of electron transport chain. So complex 1. Complex 1 also known as ubiquinone oxidoreductase, and it is made up of NADH, dehydrogenase, flavin mononucleotide, and 8 iron sulfur clusters. NADH donated from, just look at here at this point, NADH donated from glycolysis and citric acid cycle is oxidized here and transferring 2 electrons from NADH to FMN. Then they are transferred to the iron sulfur cluster and finally from iron sulfur to coenzyme Q. During this process, 4 hydrogen ions pass from the mitochondrial matrix from here to the intermembrane space, contributing to the electrochemical gradient. Next is complex 2. This complex 2 is also known as succinate dehydrogenase. So it accepts electrons from succinate. Now this succinate is an intermediate in the citric acid cycle and act as a second entry point to the ETC. When succinate oxidizes to fumarate, 2 electrons are accepted by FAD within complex 2. FAD passes them to the iron sulfur clusters and then to coenzyme Q, similar to complex 1. However, now very important point, no protons are transferred across the membrane by complex 2. Therefore, less ATP is produced in this pathway. Next is coenzyme Q, also known as also known as ubiquinone, COQ, is made up of quinone and a hydrophobic tail. Its purpose is to function as electron carriers and transfer electron to complex 3. Coenzyme Q undergoes reduction to semiquinone. Semiquinone is a partially reduced radical form, COQH, and ubiquinol means fully reduced COQH2 through Q cycle. So this process receives further elaboration under complex 3. Next is complex 3. This also known as cytochrome C reductase. It is made up of cytochrome B resic subunits containing 2 iron sulfur clusters and cytochrome C proteins. A cytochrome is a protein involved in electron transfer that contains a heme group, and heme groups alternate between ferrous and ferric states during the electron transfer. Because cytochrome C only accepts a single electron at a time, this process occurs in 2 steps, means in Q cycle. This 2-step process is called Q cycle. I will tell you later. In contrast to single-step complex 1 and 2 pathways. Now complex 3 also release 4 protons into the intermembrane space at the end of the full Q cycle, contributing to the gradients. So cytochrome C, then this is cytochrome C, it then transfer electrons means 1 at a time to complex 4. Now Q cycle. This is complex 3, and here two-step process, that is Q cycle, is taking place. So step 1 in Q cycle involves ubiquinol, that is COQH2, and ubiquinone, COQ, binding two separate sites on complex 3. So here QH2 transfers each electron to different pathway. One electron goes to iron sulfur and then to cytochrome C, while the second electrons transfer to cytochrome B and then to Q, bound at the other side. While this occurs, two protons are released into the intermembrane space, contributing to the proton gradients. So QH2 is now oxidized to quinone here, and it is and dissociate from the complex. Right now, Q bound at the second site enters the transitional COQH radical state from accepting one of the electrons. Now, second step of the cycle involves repeat of the first one. A new QH2 here, a new QH2 binds to the first site and transfer two electrons like before. Again, one electron passes to cytochrome C and one to cytochrome B, and which this time works to reduce COQH2 before it dissociates from the complex 3, and it can be recycled. Now, complex 4, this one. Complex 4, also known as cytochrome C oxidase. It oxidizes cytochrome C and transfer electron to oxygen. So cytochrome protein A and A3, in addition to heme and copper groups in complex 4, here in this group, transfer donated electron to the bound dioxygen species and converting it into molecule of water. The free energy from the electron transfer causes 4 protons to move into the intermembrane space, contributing to the proton gradient. Now, last is ATP synthase, also known as this ATP synthase, also known as complex 5. It uses ETC generated proton gradient across the inner mitochondrial membrane to form ATP. So ATP synthase contain up to F0 and F1 subunit, which act as rotational motor system. F0, a hydrophobic and embedded in the inner mitochondrial membrane, you can see here. So it contains a proton corridor that is protonated and deprotonated repeatedly as proton ions flow down the gradient from intermembrane space to matrix, right? So alternating ionization of F0 causes rotation, which alters the orientation of the F1 units. F1 is hydrophilic and faces toward the mitochondrial matrix, as you can see here. Conformational changes in F1 subunit catalyze the formation of ATP from ADP and PI. For every 4 proton means 4 hydrogen ions, 1 ATP is produced.